Protein extraction lab, take 2, sync test. The loud claps make it spike, so I can identify where it happened. Yeah, so I'm not trying to just deafen you, I swear.
No.
Okay, cool.
Alright, so the centrifugation is finished.
It's finished. We actually ran into a little technical issue where we lost some of our recording, so we are not going to run this next part in its entirety. Most of the time is just spent pipetting. We will run you through every remaining step, however.
Yeah.
Okay, so now we have pulled out our tubes once again. We've got them over here, our different sample tubes, and we have a pellet still, but we don't want the pellet anymore.
No, this has the pellet. I'm not sure if you can see it.
It probably doesn't show up, but it looks like, here's the tube, and then there's a little booger in the bottom. Yeah.
Okay.
Now that's cellular debris from when the cell split open. It lost a lot of pieces of membranes and stuff that isn't protein. All the protein should be in the solution. So this is what we want right here.
Yeah.
So now we just add it on to a 96 well plate, right?
Yeah. So we're going to take some of it and dilute it, 1 and 5,
Yeah.
which will be the D series. And then we're going to take our standards of BCA and we're going to add them to the 96 well plate the way that they were set up in the protocol.
On page 5, yeah.
And so the reason why we're doing 3 of each is just in case something goes wrong, you still have your results in duplicates. It's always good to have triplicates.
And if all 3 samples, all 3 identical samples work, then you just average your data across and you get something that's more representative of reality. So replicates are, you're going to hear a lot about replicates the more you get into experimental design.
Alright, so the reagent that we need to add, the BCA reagent.
The BCA! we know exactly all about it.
Is green. So it's green right now, and we're going to add it to all the tubes with the protein already in it.
Yeah. So what we did that was off camera is I took this pipetter, and with all of our samples and all of our standard curve generating known concentrations, I just took 25 microliters with the pipetter and I added it into the 96 well plate. All right? Now that right now is sitting off to the side working on its reaction. But once we have all of our protein now inside these wells, Bridget will add the reagent, and that's what actually generates the color that we measure with the spectrophotometer or with the 96 well plate reader.
Alright, so I'm going to add the working reagent, the copper containing reagent, to each of the tubes. I'm going to use this multi-channel pipetter. So I've got 8 tips on it, so I can go down a column all at once. So I've got it set on 200 microliters.
And there will be pictures available for you on Canvas in the module where you can see this in more detail if it's not showing up too well for you.
So solution in each of the tips, and I can go straight across or in a column.
And now Bridget has just filled up 8 wells in one fell swoop. It's a very satisfying
so
thing.
Fantastic.
Yes. Yeah.
Because if you're doing a lot of these on a given day, it's just increases the chance of getting an error.
Yep, you get tired, you get a little bored of doing all the filling, and then mistakes happen.
Yeah. So I'm going to put a white background in here. I can find a white piece of paper somewhere.
Oh, I've got the picture already that we can upload to the Canvas page.
Alright, perfect.
Yeah, so you'll see a close up there, yeah.
So this will go in at 37 degrees Celsius, and we'll see if it turns purple.
Yeah.
If we got any protein.
It incubates for about 30 minutes according to the protocol.
And actually that one's probably done now. So.
We'll take a peek right now, yeah.
Right.
We're good. Oh yeah, this looks great.
It worked?
Yep.
Yay!
We did a good job, it looks like.
Let's see.
So, our plate is now out, and we're going to see some patterns in the color. So, the more intense this purple color is, the higher concentration of protein we find.
This tension's probably good enough that we can...
Yeah, you might be able to. In fact, we might want to try to. I could try to hold it in front of the lens for a second,
Okay.
get a really nice shot of this, because this is a great plate.
And so the more purple, the more protein you have.
Let's see here. So here's the plate, and sure enough, you can definitely see the gradient in color, right? So go ahead and look in your protocol and actually look at what these concentrations are, and you'll see that the pattern makes sense going from high to low concentration, with these being all of our standard curve generating samples.
Yeah.
We will also put a picture of this up online.
And so some of the samples haven't turned purple yet, so I think it needs to go for a little bit longer.
A little longer? Okay.
But we'll send you a picture of the
Perfect.
final thing.
Yeah, you'll get it then. So that really concludes the protein extraction lab.
Yes.
Look in your protocol for the description of the assignment. Let us know if you have any problems. Get comfortable with Excel. Excel is a great tool, and you got to use it again for this lab. So yeah,
Yeah,
that's that.
and they're basically going to generate a graph like this and tell us which buffer worked the best.
Yeah, which one got you the highest protein. So you'll need to generate a graph like this. You'll also generate an equation. You've experienced this already from your carbohydrates lab. But if you need a refresher, just reach out. We're all sitting at home alone on our computers.
We want to help.
We want to help. All right. Thanks, everybody. See ya
Bye.
next lab. Bye. Cool. I think that was fine. I think we still got everything across. That took us 6 minutes. Not a big deal. So even with these setbacks, we've really only lost like a few minutes each time.